All right, looks like we are live. I want to welcome everybody to Standing for Truth. Looks like we got some good brothers and sisters and skeptics in the chat for tonight's more so impromptu show. There are several topics that I want to discuss. For example, genetic entropy. One could consider this a pre-game show to our event this Thursday. I should get up the promo for it, genetic entropy and the mystery of the genome. And so we're going to have Paul Price here for this. Sam and myself will be hosting. It's essentially a challenge to all skeptics and critics of genetic entropy. And so it is a general challenge to the evolutionary community. And we are going to engage in some impromptu debate on the topic of genetic entropy. Jackson Rowe, my good man, you're no stranger to debates. And so I'm hoping to see you this Thursday. I think it's going to be a lot of fun. I think it's going to be edifying. So I want to do a little bit of, I want to put out a little content tonight, I guess you could say, on the topic of genetic entropy, going over some of the basics, going over some of the challenges in the form of a pre-show to that event. Also, I will be recording probably tonight, part 2 to the William Lane Craig is Evolution a Theory video. I'll be recording part 2. Part 1 I released on Patreon about a week ago, and then I premiered it on the channel yesterday. And so it's great to see the feedback. It's great to see how many people have enjoyed it. I think it hit about 2,500 views in just a day since release. So if you want early access to each part in this series, make sure to sign up to Patreon. You can do so for as little as $1 a month. Part 2 should be available on Patreon in the next couple nights. And then I'll premiere part 2 probably in a week or so. I'm looking forward to the next episode or part in this one because I'll be debunking a couple of my favorite topics, pseudogenes and also junk DNA. And so the fact that William Lane Craig is looking to arguments like pseudogenes and junk DNA in support of evolution, that kind of evolution that says whales and strawberries are related through common ancestry, tells me he's not quite up to date on the technical literature regarding these issues and topics. I've really noticed that in the world of theistic evolutionism. You rarely find the theistic evolutionists engaging in debate. You rarely find, if ever, them joining the open mic debate challenges that we do frequently. And so a lot of these theistic evolutionists, unfortunately, they compromise the Bible, and they do so without ever engaging top arguments for separate ancestry. And it really is disappointing. I would like to see more theistic evolutionists at least attempt to engage some of our better arguments, again, for separate ancestry and universal common ancestry, or and young earth creation, not universal common ancestry. So I am going to post the link in the chat, especially for any critics that want to join, any skeptics that disagree with what I am going to be saying, especially on the topic of unconstrained genomic sequences and hierarchies. I want to just chat a little bit about Dr. Dan Stern Cardinal's challenge to those who hold to the separate ancestry model. And therefore, anybody in the chat who is pro-evolution and would like to join for a bit to basically engage anything that I am going to be saying, then I'm just looking at the chat, then feel free to click the link. I'll look periodically at the backstage to see if anybody is joining. So basically, this argument was presented by Dr. Dan Stern Cardinal during our last open mic. And it was an excellent open mic. I highly recommend people check it out. Jackson Rose says he'll join for Entropy. Okay. We can chat about that in a bit too, once I get onto that topic. So our last 2-in-1 open mic event, I thought it was a fantastic show. I put these on because we want to engage the best arguments that the evolutionists have to offer. I don't ever want this channel to become an echo chamber. I want us to engage the critics. I want us to interact with the challenges advanced by the critics. And so I think this series has been very beneficial to the community. It's been edifying. I learned something new every show. And we've had, over just the last month or two, we've engaged critics like Gutsick Gibbon. We've engaged critics such as Dr. Dan Stern-Cardinal. Very professional, very sophisticated open mic debate last week. And so we must have discussed for somewhere between 3 and 4 hours. The entire show went about 5 hours, as usual. We must have discussed the topic of unconstrained sequences within the genome and the hierarchical patterns that they fall out into. And so I'm just looking at the chat to see if any questions or anything like that come in that I can answer. So I'll probably be doing this for probably about an hour. And then Praise I Am is also hosting an open stream tonight. He may be focusing on other topics such as soteriology and eschatology, but we can also discuss topics related to the creation versus evolution debate as well over on Praise I Am's channel. So I'll try and keep this roughly an hour. And again, any skeptics that want to join for some impromptu discussion, feel free to do so. Okay, so let's get right into it. Basically, the argument presented by Dan, I do have one of his slides here. And so first, I do want to thank Dr. Dan for being willing to join the open mic and engage in some professional and cordial back and forth dialogue on some of these technical topics. So he made an interesting point about nested hierarchies in constrained and unconstrained regions of the genome. Basically, you have what's called conserved regions in the genome and non-conserved or unconstrained regions in the genome. And the evolutionists primarily look to unconstrained regions as being non-functional, because essentially they're not subject to purifying selection. Your conserved regions, they tend to be more similar across creatures because they are typically involved in cell processes, organismal processes that are detrimental or fundamental to the health and existence of an organism. A genomic sequence that is involved in development or involved in the heart, in lungs, in brain function. Typically, if a region of the genome like that is hit with a mutation, then that individual will be removed from the equation because it is detrimental to the health of an organism. It's big enough for selection to see. There's a lot of mutations that occur in what the evolutionists call unconstrained regions of the genome that they would argue are not under purifying selection because those genes are not important enough to the health of the organism or the survival and fitness of the organism for selection to see and do anything with. I like to point out that just because a region of the genome is not necessarily conserved doesn't mean that it's non-functional. There's a lot of regions in the genome that are involved in the immune system, cell stress responses, acting as a mutational buffer, acting in chromosomal structure and integrity, all sorts of functions that are there, but those genomic elements and sequences might be able to endure more mutation accumulation than, say, a part of the genome involved in the heart or the circulatory system. But again, just because a region is not conserved does not mean it's non-functional. Some DNA sequences may be able to tolerate error more than other sequences, or the mutations in these sequences may have functional consequences that we are not yet aware of. We are in the early stages of understanding the DNA language. We are in the early stages of understanding all the ins and outs, all the hyper technical and specific details of the embryological developmental stages, where a lot of these genes are functional and then they're shut off and repurposed a lot of the times. A lot of genes are only functional under and expressed under certain conditions. Again, a lot of genomic regions function as a mutational buffer. There's a lot of redundancy, functional redundancy in the genome. That's great design. There's a lot of over design as well. And so since we're just in the infancy of understanding the DNA language, understanding what's really going on a molecular and cellular level, it would be premature to identify a specific genomic region or a DNA element as being non-functional when the proper tests have not been done. So evolutionary logic is basically purifying selection removes new variants and keeps a genetic sequence the same. And so over millions of years, conserved regions are going to stay the same because selection has been acting upon those regions to keep them the same. Evolutionists also assume if the regions of the genome are not under purifying selection or adaptive selection, they're evolving neutrally and are therefore non-functional. The evidence suggests that there's genome-wide activity, and we can get into all that. For sake of argument, Dan says it doesn't matter if these regions are functional or not. The whole point is there should not be hierarchies in these unconstrained regions if special creation's true. Because the young earth creation model would say, okay, 6,000 years ago, God creates very specific archetypes. You have the original created kinds that are front-loaded with pre-existing genetic diversity and DNA differences. This applies within species. This applies to Adam and Eve, but also across species. And so you have DNA differences, and as a result, you have hierarchical patterns. In the biological world at creation, where humans are more similar to one creature, let's say today chimpanzees, than they are to another creature like a dog. Human designers, human engineers naturally build in hierarchical patterns. A sedan has to be more similar to another mode of transportation than it is to another. A sedan is more similar to a hatchback than it is to an airplane. And so we find hierarchical patterns in the biological world. We find hierarchical patterns in man-made designs. And so overall, the bigger picture, hierarchical patterns are agnostic. Dan's argument, and these are the kinds of arguments I believe we should engage as creationists. Creationists and evolutionists need to come together and discuss the tough questions. The questions that I would say help discriminate between the models, because there is a lot of overlap in the world of creation versus evolution. What that means is there's a lot of observations, there's a lot of evidence in this world that both models can explain. As a matter of fact, many different models can explain. Okay. But the best way to answer this question of ancestry and origins is to look to discriminatory lines of evidence or differentiating lines of evidence, a line of evidence that only one model can explain. And I would argue, and I've pointed out many times, that there are many lines of evidence that only the separate ancestry model can explain. And that those lines of evidence are typically found in the world of genetics. Honesty Angel, good question. Yeah, I did put the StreamYard link just in case somebody on the evolutionist side wanted to challenge something I was saying. I do my best to convey the other side, to reiterate their argument, If they have a problem or they have a problem with my answer, feel free to click the join link. If we go too far past the hour mark, we'll shut it down here and we'll continue in any conversation and debate over on Praise I Am's channel. And so what I can do is repost the link just for any critics in case. Let's see, I'm looking at the pseudonym says, it seems Standing for Truth has admitted Littlefoot is human. I've never admitted Littlefoot is human. What is the classified though? I've never said Littlefoot was human. I am looking forward to, I'm strongly looking forward to the end of the month. I'm going to have Dr. Peter Line on. He's a leading voice in the world of human evolution.
What's up, guys?
Jackson Rowe, good to have you. Good to have you, my man.
Hey, good to be here. I just broke my chair.
You just
by
broke
accident,
your chair.
so I'm kind of uncomfortable sitting here, but what's going on?
I blew your mind with what I was saying. You leaned back and broke your chair.
No, I think it's because I've been eating too much ice cream, actually.
Well, I'm looking forward to your upcoming debates. I'm looking forward to you joining, these future open mics for sure. Yeah. So, Jackson, feel free to stick around. Once I say something or we get into something that you'd like to engage a little bit, then feel free to jump in. I'm just kind of catching up with the chat here.
We're talking about Littlefoot. I thought I heard Littlefoot.
Yeah, I think pseudonym might be mistaking Littlefoot, Australopithecus prometheus, possibly with Homo floresiensis, which is known as the Hobbit, and possibly Homo naledi. And so I would say Homo naledi and Homo floresiensis are both human. Both suffered from a form of degeneration and pathology. A problem I have with the critics, in Jackson Rowe, you can speak to this. Okay. A problem that I have with the critics when it comes to the hominin fossil record, and at least the specific model I hold to in explaining their anomalous and atypical features. And I'm not saying you're guilty of this, but I find a lot of critics, they get confused. They think that I am saying, or other creationists like Chris Roop are saying, every single Homo naledi specimen had arthritis, or every single Neanderthal had arthritis, or hypothyroidism, or some kind of disease, right? That's not what we're saying. We're saying that the results of inbreeding, the results of existing in small populations, the effects of environment and epigenetics has resulted in these features, these atypical features, having drifted to fixation, where everybody in that population, let's say Homo naledi, okay, everybody in that population now has those traits: small body size, small brain size, curved fingers, for example. So those atypical features have drifted to fixation where they're the norm. Like Hobbit, for example, there's strong evidence there of founder effects, intense founder effects, where all the descendants of the originals now have those features. So it's not like, well, this population's all coincidentally suffering from this disease or this pathology or arthritis, right? No, it's that founder effects, inbreeding, especially in small populations, is very real. And most certainly after several generations of that inbreeding in a small population, you are going to have fixation. You are going to have evidence of inbreeding. And that evidence is negative. Jackson, what are your thoughts on that?
Sure, I don't disagree that Homo floresiensis probably had some founder effects, being bound to a single island, basically, or as far as we know. But I think the standard idea is that it's descended from Homo erectus, not like Homo sapiens. But I know you think that's also a human, right?
You don't know nothing, Jack. No, I'm just kidding. Erectus, I would argue, is a pathological form of Homo sapiens. a degenerate form of Homo sapiens. They existed in small tribes. They existed in between 50 and 100 individuals. And so in those circumstances, especially the harsh environment that they existed in, all of these different hominins, what that means is after a few generations, you're basically guaranteed to have evidence of inbreeding. And so Erectus, which is a more degenerate form of Homo sapiens, gets a small group of Erectus basically gets stuck on this island of Flores. And then that group suffers from island insular dwarfism in many ways, and reductive evolution. And so I think Homo floresiensis is a more degenerate form of Erectus. So I do agree that floresiensis, the precursor, is probably Erectus, but I would also look to Erectus as being human. Does that make sense, Jackson?
Yeah, I get what you're saying. But I mean, some people do go the route that they say it was all pathological, like pseudonyms said, that it was whatever they chewed a lot. That's why their brow ridge is really big, stuff like that. The Neanderthals. But
the problem is
that's not your opinion. But
the problem I have with creationists that just say, well, name your hominin. You know, their atypical features is because of, you know, arthritis or this disease. Well, you have to look at the babies. You have to look at the children. Do they have those same features? Naledi, for example, you know, do the babies and the teenagers, do they also have the curved fingers? Do they have the same atypical features as the adults? Well, if that's the case, that means these features are fixed in the population. If it was just arthritis, you'd expect, you know, the babies and then the teenagers and the adults to differentiate basically in those atypical features. Does that make sense? You know, you'd expect your adult Neanderthal, let's say, to have those atypical features because over time they developed them, but not the children, right?
Right, but here's something you might be able to answer. With all the Neanderthal specimens we found, they all basically have the same skeletal features. Don't you think with inbreeding there would be one or two outliers that wouldn't have so many of the same features?
Well, that's my point, is the only reason an entire group like Neanderthal, you know, I don't really like looking to Neanderthal because I think a lot of the features associated with Neanderthal are due to environment. Environment can dictate phenotype. Therefore, I believe a lot of the features associated with Neanderthal, like especially the robustness of Neanderthal, are eco-glacial adaptations. So yes, they were highly inbred. The genomic data says that Neanderthals were 40% less fit than modern humans.
Sure.
They had massive runs of homozygosity where you look at these stretches of DNA and they have identical letters saying that they basically intermarried with close relatives. Okay, but their phenotypic features, I would say most of them are due to eco-glacial adaptations. I prefer looking to groups such as Naledi, such as Floresiensis and Erectus, where, like you're saying, yes, there's uniformity in those features. And I am arguing that uniformity is because those features drifted to fixation.
Right.
So of course they're all going to have the features.
You would think there would be some specimens before all these features were fixed in the population that show, wait a minute, this one has some abnormal skeletal features that the other ones don't have.
Repeat that if you don't mind, Jackson. I'm just sending the link to pseudonym because he's not making any sense in the chat.
Let me rethink about what I said. My short-term memory is very bad. Something about, we were talking about Neanderthals. Something about, wouldn't you think we would have some bones before all these features were fixed in the population, and that shows some some variation in the skeletal structure?
If I understand what you're saying correctly, my answer is yes, and I believe we do find that. For example, with the work I'm currently doing, I think that, I'm just trying to find some slides on it. I think that Noah and Noah's family would have been closer to probably the Homo heidelbergensis phenotype, because Homo heidelbergensis seems to be the early stage of a lot of these hominins. They were robust. their bones were quite massive at times. I watched a video of Lee Berger. He was holding up this massive femur, if I remember correctly, and a little tongue in cheek, but he was pointing out like we had some gigantic hominins existing. And I think he attributed it to Heidelbergensis. And if you look it up, there seems to be a lot of data suggesting that Homo heidelbergensis, existed at 7 feet tall, some of them. And so Neanderthal that we see in terms of their genetics, the ones that are highly inbred with a high genetic load, they appear to be the end stage Neanderthal. And so the early stage could have been closer to Heidelbergensis, maybe the early stage Erectus, same thing, closer to Heidelbergensis, a more healthy phenotype. By healthy phenotype, I just mean a phenotype that's not riddled with evidence for pathology and degeneration.
Well, we're kind of back to this similar problem, because Heidelbergensis all kind of has consistent skeletal features within the species also.
Right, and that's what we'd expect, because post-flood you have this bottleneck where you have Noah, his wife, okay, and then their 3 sons, Shem, Ham, and Japheth, and then their 3 wives. And so from there, you're going to have rapid and exponential population growth where those features that Noah had will be found in descendants. Now, eventually you have subgroups that break off, right? They migrate, they become isolated on islands, for example. I like the way Chris Stringer explains it. He says, the experiment in human evolution conducted on Flores could have been repeated on many of the other islands in the region. That speculation has seemingly been confirmed on the island of Luzon. And so basically what we have in the past with these hominins is we have very rare, very atypical evolutionary, I would call them reductive evolutionary experiments going on that we don't see today. I mean, we have these harsh environmental conditions. We have just a few individuals founding a greater population, basically, where you've got intense inbreeding, intense founder effects, combined with extremely harsh environmental conditions. And the result is, as Chris Stringer says, the result of these experiments are hominins like Naledi, very atypical, like Floresiensis, where reductive evolution has basically resulted in the small brain sizes we see, the degenerative features like the curved fingers. Is what I'm saying making sense at all? If you have an objection, go ahead.
I mean, I follow what you're saying, yeah.
So, yeah, the point is, it's not like, well, okay, we have, so for example, here's Chris Stringer again, right? These are quotes from evolutionary paleoanthropologists. Notice what he says. Others, such as me, wonder whether the Luzon finds might eventually turn out to be a variant of the already known Homo floresiensis. That's what I say, is these various isolated populations look the way they do because of the conditions they're associated with. Notice what he says here. We know that island isolation can be a catalyst for some odd evolutionary changes, including, and notice this is fascinating, reversions to apparently primitive states. So he's saying reversions to primitive states as in like more Australopith-like, right? More primitive as in pre-human. But I would argue that should actually be reversions to apparently more degenerative states, or states that maybe didn't even exist before Floresiensis, before Naledi, because you're having healthy morphology, basically take a femur or brain size, and due to the conditions, due to reductive evolution, adaptive selection, you're going from a bigger brain to a smaller brain. You're going from a bigger body size to a smaller body size. You're going from straighter phalanges to more curved phalanges. He calls that primitive states. But he is saying that these isolated conditions that we see the hominins associated with, they're a catalyst for some really interesting changes or
Sure.
adaptive episodes. And but the thing is, Jackson, those would become fixed. So what you're saying is a great question, and that's why I disagree with certain creationists that say, well, these features are because of arthritis or hypothyroidism or this disease or that disease. But if that were the case, not every single individual, including the kids, would have those atypical features. No, but if through founder effects, genetic drift, and fixation, which occurs in small populations, then yes, every single one of those features would drift to fixation, and therefore those features are now the norm of that population. So if you find an individual that is categorized as Homo floresiensis, they're going to have that same set of traits. Go ahead, speak to that a bit, Jackson.
Yeah, I don't really know much about Floresiensis, but I mean, I spend my time mostly on the Australopiths and stuff like that.
Well, Jackson, you've been doing a lot on your channel. You've been doing a lot of work. Okay. I drank a lot of coconut water before I started. So why don't you just for a minute, tell the audience what you're up to at your channel, also what you have planned for the future, including any upcoming debates, anything like that. I'll be back in less than a minute and we'll continue this conversation. Jackson, go ahead.
All right. Okay. He has to go to the bathroom, so I'll take over. I noticed someone mentioned I have a snake in my picture. That's not because of some Bible reference that I'm like mocking the, Genesis or something. That was my pet snake. That was my green mamba. The government got pretty mad at me about that. I don't have it anymore, so that's what that's about. Anyway, I've been talking a lot about genetic entropy and like mostly carbon-14 lately, transitional fossils, stuff like that on my channel. That's pretty much all I've been doing. I'm going to be carbon dating some. Oh yeah, I do like exotic pets. I'm going to be carbon dating some samples actually here pretty soon as an experiment. So that'll be interesting to see what comes of it. Kind of as an answer to the rate experiments. I'll talk more about that when it comes out. There were salamanders, bubblegum, not lizards. And I saved them from being eaten. Does anyone have any questions for me about all this stuff? Like, are you guys familiar with the Littlefoot fossil? I know Donny is, but like you guys in the audience? Because like most creationist outlets don't seem to cover it much.
Jackson, I'm really looking forward to if we schedule the Robertson Genus.
Oh yeah, I got the slideshow already made up.
Geocentrism debate. I think that'll be a lot of fun.
Yeah.
So my question is, aside from your Australopithecines, okay, Dr. Peter Line and I, we are going, and hopefully you'll be there in the live chat, Jackson, to ask questions and raise some objections and challenges. The hominids, those in the human line, although you'd have the Australopithecines in the human line too, but the Homo genus, right? Floresiensis, Naledi, Heidelbergensis, Neanderthals, those that we would both agree are more human. You might say primitive human or pre-human. Nonetheless, those features, if they were to drift to fixation, as we understand is the case in instances where we have a group that exists in a small population, they're inbreeding, and the environmental conditions associated with that population are harsh, right? Think about it. Neanderthals lived in the cold, eco-glacial environments. They were highly inbred, existed in really, really small populations. Floresiensis was stuck on an island, highly inbred. They find elephants on that island that are dwarfed as well. Okay, so environment dictates phenotype and morphology. Luzonensis, same thing. Naledi found 15 individuals within a cave. Okay, so all of these examples would tell us that, okay, these atypical features have become fixed. And that's how me as a creationist would explain it. That's the model that I hold to, right? Post-Babel dispersal, many different groups migrate to less than ideal parts of the world, islands, just horrible environmental situations they exist in. And what happens is we get evidence for degeneration pathology. Does that make sense, Jackson? Do you see any major problems with that at all?
I mean, no, the way you're describing it, I still think they're different species in humans, but basically you're right about the founder effect.
Right, because that's how founder effects work. Again, Chris Stringer, he's pointing out that, and Grayson, that's a good question. That's a very good question. That's because, as Chris Stringer says, the experiments that took place on these islands, that took place with these isolated individuals, those have not necessarily been recapitulated today. Those were some very anomalous conditions. The cold ecoglacial environments of the Ice Age, these islands, we have a small group existing for generation after generation after generation. And so these phenotypes drift to what we see today in the fossil record, but have those same evolutionary experiments, as Chris Stringer says, have they been mimicked or have they been recapitulated today in the modern world? No, I don't believe so. We're not existing in the same harsh environmental conditions that these hominins did, nor are we experiencing the kind of isolation and inbreeding that those hominins did.
What about the North Sentinelese? They're stuck on an island and they look pretty much human. They are human. They look human.
Well, it's going to come down to how many generations are they stuck on that island, the environmental conditions associated with their isolation, with their inbreeding. And so I'm going with what Chris Stringer says, that yes, these kinds of isolated conditions, these types of anomalous situations are a catalyst for some very interesting evolutionary changes. And then those changes, they become fixed where everybody has it. So it's not like, well, this one individual has arthritis and then coincidentally everybody else got arthritis. No, it's those features are the norm within that population.
So Donny, we have a pretty big sample size of populations being isolated to islands. Like Jackson brought up the North Sentinelese, which have certainly been there for very many generations, but there's all Micronesia, Polynesia, all throughout the oceans. We have like a sample size that's very, very large of isolated island populations, and none of them have become anything like Homo floresiensis or Homo naledi, or like they're all still anatomically human. None of them look anything like these other, what you're saying are pathological, like Homo erectus. None of them skeletally, they all look Homo sapien. And just one other point, like when you talk about Homo erectus or Neanderthal, those were not isolated populations at all. We find Neanderthal and Homo erectus spread over multiple continents.
Appreciate those points. Grayson, welcome to the show. I'm glad that you're here because my next topic that I was going to go into is the unconstrained sequences and the hierarchies they fall out into. And so it's good to have you here because you'll be able to engage that. To respond to, firstly, for the audience, I'm pumped for your debate with CJ Cox coming up on Exodus. So you're going to be an Exodusologist soon with all the study you're doing, reading the book of Exodus. So I'm impressed. Okay, so in response to what you're saying, we'll start backwards. Neanderthals and Erectus, they existed in small tribes, but I agree with you, they were widespread. Neanderthal existing all throughout Eurasia. But each group of Neanderthal existed as a small tribe, maybe somewhere between 50 and 200 individuals. Okay, just kind of throwing a number out there, but definitely a few individuals. And that's exactly why their genetics, and I'd predict if we got the genetics of Erectus, we'd find the same thing where you have just these.
That's not how that works, Donny. You need to calculate the effective population. You don't just look at like how big their closest kin group is, because that's not how mating patterns work. You have to calculate the effective population size. It's a calculated value. It's not just based on how big the average number of people that they hung out with is.
I agree with you that we need to calculate the effective population size, but I don't believe considering that helps your case or refutes anything I'm saying. What do you believe the effective population size for, let's say, Erectus was, or even Neanderthals?
I've got no clue. I wouldn't be comfortable offering a guess, but we could certainly look it up and see if it has been calculated.
Look it up. And I would argue that whatever it is calculated as is probably a strong guess, strongly inferred, because yes, we find bones of these individuals, but we only find a few, like Naledi, we find 15 individuals, right? Well, how many individuals in total? What was the effective population size of Naledi? I mean, I think you're just guessing basically. But my point is so let's say effective population size, Neanderthals, 30,000 across Eurasia.
That's
okay.
probably pretty high even.
Okay. Well, then basically that best case scenario, 30,000, even less, say 15,000. Okay. Very small population, but that population would be broken up into tribes of, again, between 50 and let's say 100. But it's not like this tribe intermingles with this tribe and this tribe intermingles with this tribe. No, they're existing in their tribes and they're breeding, they're mating within those tribes for long periods of time.
That's not how that works. Like, and
that's
in
exactly
the actual equations, none of what you just said is even relevant. Like the effective population size, it's what's important for calculating inbreeding effects. None of the kinship group size even is a factor. And it is the case that the different tribes mingle around. Look at any hunter-gatherers or like bands of any primates, and that is the behavior that's always observed. Like, I mean, like the females or the males will go out to a different tribe, and you do have intertribal mingling. Like that's been the case throughout all of human history, and we have the genetics to back that up.
Well, you're inferring that that's the case all throughout human history. These
Neanderthals.
Wait. Listen. The Neanderthals existed in conditions that are totally different to conditions we live in today. The cold ecoglacial conditions they existed in, the highly inbred nature of them, and then existing like that for generations and generations and generations. Okay, whatever inbreeding effects take place, those will be manifested outwardly, and eventually they're going to drift to fixation. Again, I was telling Jackson earlier that Neanderthals, I don't think are the best example, because I think a lot of their phenotype, at least, is the result of cold ecoglacial adaptations. But somebody like, a group like Floresiensis or Naledi, yes, I would argue that isolation, inbreeding, and founder effects is what explains their atypical features.
Here's something else about inbreeding. It's kind of brings out recessive traits kind of randomly. It seems like with axolotls, look at those. You got leucistic ones, melanistic ones, albino ones. I mean, they don't really look skeletally; they look consistent. But I mean, you see what I'm saying?
Yes. So I did. Let me post the link one more time, because there are. Oh no, it looks like pseudonym. I thought he was going to be joining. So let me post the link one more time. Anybody that wants to join, feel free to do so.
He said in his comment that one day he'll be as rude as me. So,
yeah. Well, what I respect about Grayson is he's bringing up objections, pseudonym, in the live chat, but then he's joining to discuss them. But a lot of what you're saying in the live chat, pseudonym, I love you, but it's not really making a lot of sense in terms of at least how you're writing the objection. So feel free to join. That's what this is for. Praise I am. Good to have you, brother. What's going on?
What's up, my fellow homos? Not pretty good, man. Just listening and learning, learning science.
Your fellow Homo sapiens.
That's right. I'm a Homo sapien, bro.
Homo sapien and proud. Well, it's good to have you, Praise. I know eventually we're going to be transitioning to your channel, so good to have you. Feel free to jump in on any of this.
Appreciate it.
So here's what I was going to talk about today. I think the genetic entropy portion we'll just save mostly for our Thursday open mic.
Oh, man.
But you're looking forward to that, Jackson. I know you are, and so is Grayson. So unconstrained sequences and hierarchies. I thought the last open mic was excellent. Grayson, good to see that you uploaded a large portion of it as well. So nested hierarchies in constrained and unconstrained regions. Apparently, it's a direct refutation of the common design hypothesis. I had a lot of fun discussing this particular issue. So Grayson, Jackson, anybody who wants to discuss this particular argument or challenge, you're saying that if, according to the separate ancestry model, the original created kinds are designed in a way that allows them to have internal diversity. As a result of internal diversity and basically DNA differences that are separating species and kinds, you are going to naturally find some kind of hierarchy. From that position, you would argue that over 6,000 years, more accurately 4,500 since the flood, since there was a bottleneck, those hierarchical patterns that were once there in unconstrained sequences, they should no longer be there because mutations accumulate in unconstrained sequences without any real selection. There's no purifying selection acting upon those regions. And so those regions should diverge more and more where there is no hierarchy seen if separate ancestry is true. Go ahead, speak to that. Is that more or less accurate?
Isn't there background selection, though, that selects against the ones that are harmful, but then also parts that are not really doing anything?
Well, great. Grayson,
I mean, you could have some
go ahead.
selection effects just if they're close to like areas that, like, as a chunk, you can have selection effects in chunks of the genome. But largely speaking, in unconstrained sequences, there's not going to be purifying selection. So you would expect a pattern of mutations to be pretty stochastic, which is like kind of synonymous with mostly random.
Okay. My challenge is this, but also a question of curiosity. And in our discussion during the open mic, I don't feel like the evolution side adequately answered it, but I do really appreciate the technical back and forth. And so this is probably up for debate, right? So let's see. I don't believe that the evolutionary model does predict hierarchies after millions of years in unconstrained sequences, because these unconstrained sequences, there's no purifying selection, or there's very limited, as Grayson was saying. Remember, selection sees the really big mutations. Those are the ones that it sees. It can amplify the best beneficial mutations, but largely, and what we see in unconstrained sequences, largely what we see is an accumulation of neutral mutations. And so they're not subject to selection. And so they build up over time and they drift through genetic fixation, randomly for the most part. And some drift to fixation. Anyways, over millions of years, if we have all these mutations accumulating generation after generation without selection in these unconstrained regions, then today we shouldn't find any consistent hierarchical signature, because those mutations should have scrambled or muddled the unconstrained sequences to the point where there is no hierarchy. Can you speak to that, gentlemen?
Yeah, I can pick that up real quick. I think it's a basic question of numbers. So you and Matt like bringing up that CO1 gene study, right, with the DNA barcoding. I just want to point out, because I didn't get to address it in the last open mic, but I just want to address that is not talking about the sequence. That's talking about barcoding, which is like different things. But in that study, you point to basically the conclusions that they have, that That for most species, the CO1 gene kind of looks different to barcoding after like 200,000 years for like over 90% of species, right? So that CO1 gene is less than 700 base pairs. So basically you're kind of starting to get a different barcode after like 200,000 years for 700 base pairs. So then if you're talking about the total unconstrained regions, you're talking about billions of base pairs. So for 700 base pairs, it takes 200,000 years to get a different barcode. So for it to lose that pattern of nested hierarchy, I mean, just do the math. I mean, for 700 base pairs, it takes 200,000 years.
Right.
How much would it take for billions of base pairs?
So a 200 base pair region. Just a second. My wife's bringing me a bottle of water. It's right here.
Yeah,
Donny drinks a lot of water.
it's not perfect because it's DNA barcoding, and it's not
Right.
really it's not really looking at whether or not you can reflect nested hierarchical patterns off of that, but just as a proxy to gain an understanding, you can look at that CO1 gene study. And
That's a
for
good proxy.
700, what?
I agree. That's a good proxy. That's
Yeah,
a good,
for 700 base pairs, you get a different barcode after about every 200,000 years.
Yeah, that's
So
a good
then-
control.
Yeah. So then if there's, estimates are anywhere between 80 to 90% of the human genome is non-constrained. So that's out of what, 4 billion? So that's going to be only like, 3.5 billion, let's just say, unconstrained base pairs. How long until you get until the nested hierarchical pattern disappears from that? I mean, it would be many billions of years.
Okay. Before I give you a strong rebuttal to that, I feel like I need to ask you a few clarifying questions just to make more sense of this argument. Unconstrained. What's your definition of unconstrained or unconserved?
Like if it's not under if it's not under the effects of purifying selection, like basically if that the actual sequence of the base pairs is not contributing to the function of that region. So if there's an equal chance of that base pair position being any of the 4 letters, like, then it would be unconstrained, like in a population.
How do you know if a region of the genome is under purifying selection?
I believe that there are genetic analyses that can be done to estimate if a region is constrained or unconstrained.
Are you looking at the amount of mutations that are accumulating in one region versus another?
Yeah, I think you compare, like, in a given population, certain areas, and like certain, you like look at certain base pair areas, and you can tell like how constrained or unconstrained they are based on the variance within a population. That's my understanding, but they could estimate them through other means. I'm just not sure.
I suspect some inherent circularity in defining unconstrained, because I hear evolutionists like you saying, well, unconstrained is a region of the genome that's not under purifying selection. But then how do you know if an area is under purifying selection? Well, because it's unconstrained. I feel like there's a circularity in the definition of it. Speak to that, Grace. I'm just trying to really clarify some of these points, but that's kind of what I'm gathering from the basic definition of it. It's unconstrained because it's not subject to purifying selection. How do you know it's not subject to purifying selection? Because it's unconstrained. It doesn't have as many mutations accumulating basically as compared to another region, it has more mutations accumulating than another region, which doesn't. Something for the heart or something. So speak to that briefly, Grayson.
I would need to look into specific studies of their methodology for how they're like operationalizing constraints. But I do know, like I just sent a study in the private chat that is like a genome-wide constraint map of the genome that they looked at for 76,000 human genomes. So I believe that they take the population, like a given population, like in this case, 76,000, and they compare each and every position and look at the variance in the population. And the closer you are to having, basically random positioning of letters at certain locations, right? And this, remember, it's important to keep in mind that we're talking about individuals here, and genetic drift culls like 99% of individual mutations. So when you're talking about the patterns of nested hierarchy in non-constrained regions, you'd be talking about like more fixed, which is a population size effect. So when you're looking at individuals in a population, you can compare each individual region and see like the closer that they are to being, 25, 25, 25, 25% of each of the four letters. That means that it's closest to being non-constrained because it doesn't seem to have any impact which letter is in that position.
Okay. Now, how much of the non-coding regions? So basically we have what, 2% of the genome codes for proteins. Would you argue that most, if not all, of your protein coding regions of the genome are constrained?
Those are the most constrained typically in the genome, are the protein coding regions. But the regulatory regions, any sequence that's where the sequence is important for its function is going to be more highly constrained.
Okay, so you would then argue, I'm guessing, a lot of your regulatory regions and so although the non-coding regions of the genome are more unconstrained, there are some areas within the non-coding regions of the genome that are constrained, like regulatory regions.
Yeah, that's an important point. So non-coding is not synonymous with non-constrained. They don't mean the same thing. So yes, you can have a non-coding region that is highly constrained.
Okay. Those clarifying questions help. So let me bring up a point here, and it's also somewhat of a counterpoint. So in order for forward evolution to work over millions of years, the neutral theory of evolution, don't you require enough mutations, neutral mutations over time to drift to fixation in order for enough diversity basically to accumulate in organisms for serious evolution to take place? You have all these neutral mutations accumulating. Eventually they reach fixation and evolution progresses.
Well, if they're neutral mutations, then they wouldn't really be contributing that much to any fitness. But I mean, sure. I mean, genetic drift does happen, and genetic drift can fix neutral mutations for sure. It does.
So if you have, just throw a number, say you had 50, let's say the per generation neutral mutation rate was 50, right? As Fatuma says, since on average it'll take 4 NE, basically how long it takes for mutations to drift, neutral mutations to fix to be fixed in a population, generations for such mutations to reach fixation, about the same number of neutral mutations should be fixed every generation. In the evolutionary scenario, you have mutation drift equilibrium. Okay, but for the young Earth creation model, for the audience sake, because Grayson, I know you already know this, we don't have enough time to reach an equilibrium where we have a lot of substitutions taking place. I'd argue, yes, after the flood, you have 8 people. And for, somewhere between 500 and 1,000 years, the human population doesn't spread out to all parts of the globe. So you can have some fixation there. But since that's spreading out, you've got no fixation. It's too slow. People are spread out. So, but in an evolutionary model and time scale, you have mutation drift equilibrium. And therefore, with the longer time scale, the number of neutral mutations per generation that accumulate, that same number equals your fixation rate. So say we had 50 neutral mutations per generation, the per generation rate, you'd also have 50 mutations becoming fixed per generation. So that would as a result, over millions of years, muddle this hierarchical signature more and more, because that's a lot of mutations that are becoming fixed. When a mutation gets fixed, it's now stuck in place where every single individual in the population has it. If genetic entropy is true, and we're going to talk about this in great detail on Thursday, and those neutral mutations are more so nearly neutral, as in they're slightly deleterious, that's actually irreversible, unless you get a back mutation, but that's pretty rare, but irreversible damage in that population, because that nearly neutral mutation is now fixed where everybody has it. There's no going back. It's not going to be removed by selection. It's not going to drift out of the population. And so, yeah, I would argue this problem, if there is a problem, is a bigger problem for the evolutionary model because of how many mutations would become fixed per generation in an evolutionary time scale. Speak to that a little bit, Grayson.
Yeah, so I think that you brought this up with Dan on the last stream, and he pointed out that this is sort of an idealized math right here, and it's for haploids, and it's assuming like random mating, or there's all kinds of assumptions baked into that statement from the introductory textbook there. So I can take it as like an approximation, but again, it's not going to be very exact, because those assumptions that go into that math are obviously not true. So,
but is this theory true or not? Like, do you agree with this basic population?
I just addressed that.
I said that it sounds like you're saying, well, it's true, but it's not really.
It's an approximation, but it's like not going to be, it's not going to be accurate in real life, because we're not haploid. Recombination happens, non-random mating happens. There's a lot of things baked into that math in the assumptions that it rests on that are just not in reality true. But it can be a fine approximation.
Well, I would contest that
it's
it's
a larger
only
problem, even if it's all true and it's exacting. I think that it's still a problem of not understanding the sheer numbers involved. I mean, how many neutral mutations become fixed each generation out of 4 billion base pairs? It's not going to be that significant when you're looking at the total numbers.
Well, so if the rate of fixation of neutral mutations equals the neutral mutation rate, if it's 50, if it's 100, if it's 10, that's how many are becoming fixed each generation. But over the course of a million years or millions of years with the evolutionary time scale, that's a lot of episodes of irreversible or nearly irreparable damage, where basically these
neutral.
Right. And that's the question. Are they completely neutral, or are they nearly neutral where they're only slightly deleterious? I'd argue if they're nearly neutral and slightly deleterious, slightly harmful, then that makes evolution impossible.
Yeah,
you're going to have to argue that they're completely neutral. There's no deleterious effects.
So what's that? What you're saying is called negative epistasis, which means that all of these slightly negative effects add up to be a large negative effect. And that's not observed. Like, we haven't observed negative epistasis happening. I mean, it's just something that we don't observe. These seem to be, for all of our observations, neutral mutations that are not adding up to have some. When you have enough neutral mutations, suddenly you have a big negative effect. Like, that doesn't seem to be happening in real life.
But I feel like there's a few points here that I want to address, though. Before I ask you this, I want to point out that Dan, Dr. Dan, and again, I appreciated his time during that discussion. I thought it was excellent. When I showed him the quote, he did say, okay, it applies to diploid organisms, but the math is complicated. And so this is the theoretical basis of the molecular clock, as Fatuma says. But when you start accounting for various things, then it becomes more multi-dimensional. Okay. But then when I asked him if the math has been done specifically on humans, he said he hasn't done it, and I haven't seen anybody that's done it yet. So I don't know how impressive this evolutionary response is, that is basically, well, yes, this is true, but when you actually do the math, it's more complicated. Well, where's the math? Where's this non-existent math that makes it more complicated?
Well, you're taking this from like it says right there on the screen, an introductory level textbook.
Yeah, it's
Why don't
basic
you go-
population genetics, right?
Yeah, why don't you go to a graduate level textbook and see what it says there?
Well, because this is like, for example, when evolutionists say, "Oh, well, I mean, you're looking at the per generation mutation rate. You're supposed to look at the fixation rate." It's like, dude, basic population genetics says that your per generation neutral mutation rate equals the fixation rate. So if it is 50 per generation, then 50 are becoming fixed per generation in an evolutionary time scale. I would argue that that time scale isn't there, but if you want that time scale, you do have this problem, especially if these neutral mutations are actually slightly deleterious or nearly neutral.
How is it
which
a problem, though?
Because if you have 50 slightly deleterious mutations being fixed every generation, then that's irreversible or irreparable damage. I understand there's back mutation, so let's say nearly irreversible damage in the genome. That damage is now stuck in place. There's no way to get rid of it unless that population goes extinct. But that's extinction now.
Well, individuals can go extinct. Will die, not extinct is the wrong word. But
well, I wanted to ask Grayson this: how do you interpret Kimura when he says under the present model, effectively neutral, but in fact, so to me it sounds like he's clarifying what effectively neutral means. Very slightly deleterious mutants accumulate continuously in every species. So when Kimura uses effectively neutral, isn't he defining that as being slightly deleterious?
I don't think so.
Well, then what is... I mean, he says, under the present model, effectively neutral. What does that mean? In fact, very slightly deleterious mutants accumulate continuously in every species, and here's the rate of loss of fitness. Per generation may amount to, 10 to the 7 per generation. And so, yes, if 50 effectively neutral or nearly neutral mutations are accumulating every generation, and that amount are also being fixed in the evolutionary timescale, that is irreversible, irreparable genetic damage population-wide.
Here's something I don't get about this. They have to be neutral or nearly neutral to accumulate unseen, but as soon as they become harmful, they're going to be selected out.
But then they're not neutral anymore. But then they're not neutral anymore, so it doesn't apply.
But I'm saying as soon as it causes a bad effect, and it's going to cause a bad effect in all of the population at once, it would cause a negative effect in an individual with a hypothetical.
And that's a great question. That's the challenge. That's the problem, is selection's so limited because selection involves the death or removal of individuals. And so if population wide, you have 7 billion people that have accumulated all these nearly neutral mutations, and every generation basically is worse off than the previous generation. Okay, like you said, enough mutations can accumulate where the damage is large enough for selection to see. Well, what are you going to do, Jackson? Are you going to just remove the entire human population? That's extinction. Well, how much of the population is going to be removed?
Not all humans have this. Humans don't have all the same mutations. So, that totally changes the equation, right?
Every generation, you have roughly 100 new mutations per person per generation.
Right, but not all the same.
You have 7 billion people. That's 700 billion new mutations this generation alone. The human population has experienced every possible mutation in one generation.
Right. I was reading a paper, and I don't have it. I don't remember the exact title. I have to pull it up, but I have it somewhere. It said deleterious mutations accumulate faster in bigger populations, but their mean effect is diminished, and they're more effectively removed.
Right, but that's the thing, is by the time a population, okay, another way to call this is carrying capacity or even mutation count mechanism. By the time a population reaches a point where the genetic load is so extensive that Mother Nature selection can now see it, that population has descended into such genetic sickness that the only way selection can save the day is basically extinction. But extinction's not good.
Here's what I don't understand about that. What I'm saying is not all humans are going to have the same mutations. The same mutation rate, yes, but not the same mutations. So as soon as something negative builds up in one, maybe that individual would die, and a more fit, so to speak, genome person would continue to live, and it would just kind of reach an equilibrium, right?
The problem with that, Jackson, is it's a misunderstanding of how genetic entropy works. The point is, yes, there is fitness variation in the human population. Okay, some individuals have more deleterious mutations than others. But it's that every single generation as a whole is more mutant and more worse off than the previous generation. Like me today, you today, we're more mutant than we were yesterday. We're more mutant than we were 10 years ago. And so, yes, there is fitness variation, but nonetheless, population-wide, every single human being is multiply mutant, and every single human being is more mutant than the previous generation. And so if you were to invoke, for sake of argument, unrealistic selection, you removed 50% of the population, but it was only 50% of the worst mutants, those that actually have the most deleterious mutations. So you got rid of, say, 4 billion. Well, you're still left with 4 billion people that are more mutant than the previous generation. And so even though you've removed the worst from the equation, those that you're left with
All right, let's say
are
that
still
happened.
worse than the previous generation. Does that make sense?
Let's just entertain this and say that happened. 4 billion people with horribly harmful mutations died, and 4 billion people with pretty much neutral mutations lived. And then the population would get back to that level, 8 billion again. And then the process would repeat again. And that kind of reaches an equilibrium.
That's a great point. But the problem is, a great point to engage, that is. So you're left with the 4 billion people that are still more mutant than the previous generation, right? Well, now that population continues to grow where you get another 8 billion people, however many years that takes place. Well, those individuals now, say 100 years later, whatever it is, they're still way more mutant than the generation was 50 years before that, or 20 years before that, or 10 years before that. So yes, selection can certainly slow down the process, but it's never actually going to solve the process, because again, every generation will be more mutant than the previous generation. And eventually a population will descend into such genetic sickness where you're basically just going to get a mutational meltdown. It's tough because eventually a population reaches a point where it kind of it becomes more of a, not that it's less of a problem, but that population can stay in that condition for, an extended period of time, but they're staying in a condition that is essentially mutant.
Donny,
does that make sense?
Screen?
Yeah.
So I would just want to point out that what you're saying is not true if there's such a thing as neutral mutations, effectively neutral mutations. And I found the rest of that quote, as you can see here. As you can see here. Okay, my computer's kind of okay. This is the quote in its context. So first of all, I want to point out that he's not talking about all effectively neutral mutations here. Like when you read it in the context, he's talking about different issues that could potentially arise. And again, this is in 1979. Further work has been done. But in this original paper, he's talking about there could be effectively neutral mutations that are in fact very slightly deleterious, that can slip under that threshold of being visible to selection, right? So he's not talking about all effectively neutral as you're trying to make it sound, like when he's saying that you're trying to make it sound like he's saying all effectively neutral mutations are in fact very slightly deleterious. He's not saying that. It's not.
Well, no, I'm not saying. I'm saying when he uses the term effectively neutral, that's what he means.
But that's not what that is not the case. That's what I'm saying. That is not the case.
Well, where does he say that?
Have, under that big umbrella of effectively neutral, you can have very slightly deleterious mutants. That's not to say that all effectively neutral mutations are very slightly deleterious. He's not saying that. Like, he's saying that these very slightly deleterious mutants can fit under that umbrella of effectively neutral and kind of smuggle in, which is what you're saying, that this can build up. That's what he's talking about here.
Well, and
then
that's
you have to read until the end where he says that whether this small rate of deterioration in fitness constitutes a threat to the survival and welfare of the species is a moot point, but it can easily be taken care of by adaptive gene substitutions. Like, he answers the problem right there. You just only were quoting the first part.
Well, I appreciate you pointing that out, but I'll go back to the quote that I had. I have that part in there, and notice here. Oh no, wait, right here. Whether such a small rate of deterioration in fitness constitutes a threat to the survival is a moot point. This will easily be taken care of by adaptive gene substitution. So he's saying it can be, but he never actually did any work. He never provided any math. He never ran any simulations to.
Do you know that for a fact?
Demonstrate. Yes. He never examined this argument or this assertion basically comprehensively, but also.
Do you know that for a fact, though? Like, are you just saying that he didn't do that in this paper, or you're saying throughout his whole career? Because he's a very established geneticist.
Well, where
You're
did he?
saying that he never did that. He never constructed any mathematical models showing that to be the case.
I'm not going to go on record and say that he never did, but I predict that he hasn't, because I haven't seen anything presented where he ran a sophisticated analysis or he provided any evidence backing up this claim that adaptive gene
have you
substitution.
specifically looked for the evidence?
Yes, yes, I have. Yes, I have. And so, and it's not found. He makes an assertion, and maybe he's hoping other population geneticists actually look into that, but no sophisticated analysis of this assertion has been done. I can be corrected if somebody presents me with.
I definitely would like to hear Zach Hancock see if he agrees with you on that.
Okay, but I wanted to point out a lot of this as well depends on how much of the genome is functional, because I have a feeling Kimura is saying yes, those effectively neutral mutations that are very slightly deleterious are those that are occurring in, say, a non-coding region of the genome that's maybe functional in regulation or something. And so they're going to have to maintain, I believe it was Kondrashov as well, who argues the genome is no more than 10% functional, in order that the majority of the genome can actually absorb these neutral mutations, making them absolutely neutral and not slightly deleterious. But if more of the genome is functional, say 50% of the genome is functional, well, that's now 50 nearly neutral mutations accumulating every generation, and not just neutral, as in with no effect on fitness. I mean, Iyer, Walker, and Kitely here says this: it seems unlikely that any mutation is truly neutral in the sense that it has no effect on fitness. All mutations must have some effect, even if that effect is vanishingly small. I mean, yeah, I'd agree with that. All mutations must have some effect on fitness. Now, whether he's referring to functional regions of the genome, let's say in the non-coding regions, speak to that a little bit. But I would agree that, yeah, no mutation can be considered absolutely neutral in the sense that it's got no effect at all. No, I would argue that each mutation, most of them having only a small effect.
Yeah, but what do you mean by small effect here? Because we've talked about this exact thing before, and I mean, if you read the rest of their paragraph, they talk about what they mean by effectively neutral, and it's totally fair. But if you really wanted to consider like some sort of, if you wanted to look, I mean, they even say vanishingly small, not just small, vanishingly small effect. So if you really wanted to consider like on a minute scale the impact of fitness of every single mutation, well, obviously to produce each letter of the gene of the DNA, like the A's, the C's, the T's, the G's, is going to have a slightly different biochemical cost in terms of the amount of ATP and resources it takes to create like a purine or pyrimidine. Like, these are different chemical compositions. So obviously they're going to have different energies involved in creating them. So even if you just have a mutation from like, an A to a T, well, suddenly you've changed the total amount of energy cost by a very, very, very small amount, but a change. So yeah, obviously you can calculate that it has an effect on fitness, but it's so small that it's negligible for all intents and purposes.
I would argue that a single spelling mistake in a book the size of an encyclopedia, that is vanishingly small. But enough of those typographical errors or spelling mistakes accumulate over time, then that book is no longer readable.
Right, but
Okay? And
if 90% of that encyclopedia is not sequence, like it doesn't need to be in a specific sequence, like an unconstrained region.
right, see? And so it comes down again to junk DNA. If the majority of the genome is junk, let's say only 10% is truly functional, then yes, you're going to say that the majority of these mutations, say the mutation rate's 100, only 10 of those are going to be nearly neutral or effectively neutral, as in slightly deleterious, because the rest of them are landing in positions in the genome that's just neutral junk. And so it has no effect on fitness. So a lot of this rises or falls on junk DNA. Now, I will say Mendel's accountant, which I understand you're not a big fan of,
Nope.
but they've at least run simulations where they assumed that the majority of the genome was junk. And the results were that even if the mutation rate was somewhere between 1 and 5 per generation that were nearly neutral, then genetic degeneration was inevitable. So it appears that even if the genome's 10% functional, genetic degeneration is still inevitable. But
Yeah,
again, that's going to rise or fall on a debate dealing with Mendel's accountant.
So
but
two things. One, the consensus among geneticists is that junk DNA is very real and exists and can be demonstrated to exist. That's the consensus among the experts in the field of genetics. Two, speaking specifically about Mendel's accountant as a model, I want to just ask you to be skeptical, and I want to ask you to look at the assumptions that go in to the math of Mendel's accountant and ask yourself if those are reasonable assumptions to make.
okay, I want to point out a couple things. Since we just argued extensively about junk DNA, we're gonna leave that for now. Okay, obviously you believe most of the genome is junk. I would argue that most of it is functional, and we just engaged that for a while the other day. But going back to the unconstrained regions and the hierarchies that they fall out into, okay? My first point, I would contest the claim that this is predicted by evolution, right? You've responded, you've given some points to consider. But the points that you've made equally apply to the creation model, because firstly, our time scale is only 4,500 years. Those unconstrained regions comprise a lot of DNA base pairs. Yep. They're massive. Again, only 2% of the genome is protein coding, 98% is non-coding. I understand not all of those non-coding regions you'd consider unconstrained, but most is what I remember you saying. And so
it's like 80 to 90% of the genome is non-constrained.
Okay. So in 4,500 years, we're not going to have substitutions. The neutral mutation rate is not necessary. We don't have the long evolutionary time scale for there to be this mutation drift equilibrium where you basically have your neutral mutation rate equaling the fixation rate over time. So we don't have those instances really where you've got irreversible damage, where you have all these mutations becoming stuck in place or fixed. Yes, after the flood, I do believe we have rapid fixation during that time period. But once people spread out to all parts of the globe, well, the genetic fixation is going to be very rare. So my point is, we only have 4,500 years for mutations to accumulate in your unconstrained regions. 4,500 years is nothing. You're not going to get a significant alteration in those hierarchies that were there at creation. You might get some change, but you're not going to get a whole lot of change.
Have you seen that study on the substitution rate measured at Tristan da Cunha, an island in the South Atlantic that was settled by the British only a couple hundred years ago? Like maybe it's 300 years that, and they've measured the substitution rate, and it matches the phylogenetic substitution rate. like measurable, and we know when the first people showed up on that island, only like max like 300 years ago, and we've measured the substitution rate in that population. It's a small population.
Right, but I'm not. Are you trying to say that because that's 200 years? In evolutionary time scales, you'd have your neutral mutation rate equaling the fixation rate.
Donny, are you saying there's not enough time for fixation to happen?
No. In the evolutionary model, you have the long times. You have millions of years basically for mutation drift equilibrium, and so therefore your rate, your per generation rate, where neutral mutations accumulate, they will equal your fixation rate. So if you have 50 per generation, you have 50 becoming fixed, basically. Okay. But and remember, those unconstrained regions, there's basically no selection. Grayson and Dr. Dan argue limited selection. But the whole argument against the creationist model depends on those unconstrained regions not being subject to purifying selection. But when I point out this challenge that there shouldn't be a hierarchical signature, if the evolutionary model is true, now suddenly different forms of selection are invoked. It sounds like the evolutionists
not true.
Cake and eat it too.
No,
yeah,
like
well,
the
Dr.
objection.
Dan was looking to the linkage blocks. Remember, he said if a deleterious mutation accumulates in one of these large linkage blocks that have accumulated neutral mutations over time, now selection removes that organism from the equation.
Yeah,
That's
he's
selection.
just trying to be careful, like with his statements. Like the entire, like for the majority of neutral mutations,
right?
Like it's probably not going to be the case. I mean, for the majority of mutations, they're going to be neutral and they're not going to be under selection in non-constrained regions. That's for the vast majority. There are always exceptions. This is biology. There are literally always exceptions to everything you learn in biology.
And I agree, the exception doesn't overturn the rule.
So Dan's just trying to be careful of making absolute statements in biology when there are always exceptions. So he's a scientist. He's trying to be accurate in his statements, but that doesn't mean that you can say with any supporting, there's no support to say that in evolution we would not expect for the non-constrained regions to show a nested hierarchy. That's absolutely
I'm not okay.
Prediction.
I understand what you're saying, but I don't think my point's being heard. The point is, if over millions of years you have unselectable mutations accumulating every single generation, unselectable meaning they're not subject to purifying selection. Okay, if that is not going to alter the hierarchical signature, then 4,500 years since the flood of mutation accumulation in unconstrained sequences is also not going to alter the hierarchical signature. Those hierarchies that were there at the flood are still going to be there 4,500 years later, even if several mutations have accumulated in unconstrained regions of the genome. That's my point.
But the hierarchies of the mutations wouldn't have any reason to be there.
There's not. It's 4,500 years of mutations in a model that doesn't have mutation drift equilibrium, where you basically have hypothetically 50 nearly neutral mutations accumulating per generation, and therefore 50 being fixed per generation. We don't have that issue. And so, 4,500 years of mutations accumulating in regions of the genome that comprise how many millions of DNA letters? It would take a long time to alter. Right. Yeah, exactly. So it would take a long time to alter those regions to the point where there's no longer a creationist hierarchy. But I mean, the whole point of incomplete lineage sorting, the whole point of examining various different gene trees, right? Depending on what you look at, gives you a totally different tree. Aren't you arguing that, over time, mutations have accumulated in a way where, depending on the gene you look at, especially in unconstrained regions, might actually put, a human and a gorilla closer to a pig than to an orangutan? I'm just throwing an example out there.
Like, you have to look at the total. You can't just look at one or two genes
exactly right
and make your comparison. You have to look at a broader amount than just looking at one or two genes.
Right. And you're doing that because mutations have accumulated and accumulated at different rates in different people groups, or different organismal groups have had different histories, different patterns in terms of mutation distribution. And so you have to look at a larger repertoire of genes rather than just cherry picking a single gene that may have mutated differently across the biological world. And so now you have an inconsistent hierarchy. You're saying that's not enough. You got to look at the bigger picture, right? You got to look at a big set of genes.
Yeah. When you look at the big set, then you start to see the phylogenetic relationships.
And that's my whole point. My whole point is the creation model doesn't have this struggle or this challenge, because we only have 4,500 years of mutations accumulating in these unconstrained regions that number in the billions of DNA base pairs. There's not going to be a massive alteration in hierarchies, is my point. I don't see it as a challenge, especially if it's not a challenge to the evolutionist after millions of years of unselectables accumulating and building up over time. Does that make sense?
It would make sense if we didn't have instances of like pseudogenes or ERVs being broken by mutations in the exact same place.
Well, where you find an ERV or a mutation, or pseudogene, I should say, that has, genetic mistake that's shared between humans and, let's say, chimpanzees, you're assuming that DNA difference is the result of a mutation to begin with. That's the first assumption. Creationists don't believe that all DNA differences are the result of mutations over time. That might actually represent the functional specificity of that specific organism. Maybe whatever that is that you're interpreting as a mutation is there for a functional reason, something for the human, something for the chimpanzee. So you don't necessarily know that is a mutation. Maybe it was designed that way.
So what is the design of us not being able to synthesize our own vitamin C so that people get scurvy? What's the design basis for that?
Well, that's one simple example. That's the GULO pseudogene, right? I would argue that there are some creationists that argue that is the way it is for a functional reason, but not yet known, because humans and primates can, humans and the great apes can synthesize their own vitamin C. And from the creation model, we were created in a garden where we could eat the fruits, the vegetables, the seeds. And so we're getting all this vitamin C externally from the environment. And so the question is, do we actually need a gene, therefore, to synthesize internally vitamin C?
Well, tell that to all the people. Tell that to all the people who have died of scurvy. I mean, right, it was just part of God's plan that they didn't have the ability to synthesize their own vitamin C that would have saved their lives.
Well, they existed in scurvy. Wasn't this have to do with sailors existing for long periods of time on the ocean, not realizing that they need to eat fruit?
Yes, scurvy is common among them, but lots of people throughout history have died of scurvy. I mean, it would be real useful to them to be able to produce their own vitamin C.
Right. That's the point of the fall, is Adam and Eve were created in the garden in a world that was 90% green, where you're not going to be subject to the harsh realities of scurvy. But especially after the fall, after the flood, yeah, you're going to have conditions now where you're not going to get enough vitamin C, and it's going to affect you negatively. But I'm just saying, maybe we never had a gene for vitamin C. That's a hypothesis.
If we never did, then it's just super weird that we would have the gene in our genome to produce vitamin C, but it would just be broken in a very specific location. And you're saying it was broken from the start in this location to look like it used to be a functional gene. Like it is a copy of the functional gene. It's just broken and corrupted in the exact same way that it's broken and corrupted in our similar, primate cousins.
Well, this is the
let me interject real quick.
Hey, let me just, T-Rock, I want to say this, then you jump in, because I don't want the audience to think that I'm making an argument that I'm not. Firstly, there's two parts to this argument, right? There's an aspect of homology, as we share this similar gene with the primates, and we also share this similar gene with other creatures like mice that actually has a function to it, which is to synthesize vitamin C, correct? But ours is broken. We can't do that. Neither can a lot of your great apes and other primates, right?
Yeah.
Okay. So there's so it's two-part argument, homology. So we could say, well, common design. But the other part is, but wait a minute. Our version of that gene is broken. And it's also broken in much the same way in chimpanzees, but in an organism like a mouse, it's functional in producing vitamin C. Well, that's the point, is the aspects of, let's say, the human GULO gene, and then also the chimpanzee gene, the aspects that you're looking at to say that it's broken might actually be there for its particular function. And maybe we don't know what the function is because we've always just assumed, well, this is just a broken vitamin C pseudogene. Nothing to look at here, folks. It's a prediction. I'm not necessarily saying that that's the argument I hold to, because I do have a paper. For sake of time, I'm not going to pull it up right now. It's a really interesting paper. It's a really comprehensive paper, and it goes into all the different creatures in the biological world that have actually endured damage to their GULO gene that synthesizes vitamin C, to the point where many within the biological world can no longer synthesize their own vitamin C, and that gene was broken independently. And so there seems to be a hot spot there, basically, where it can break independently.
But it's not broken in the same spot as it is with us and chimps and other
Okay,
primates.
let me address that real quick. So Dan made is my audio coming in okay?
Yes. T-Rock, you take over. I'm going to run to the restroom.
Okay. So Dan made a big point about mutations being probabilistic. So what that means is, for any given mutation, there is a specific probable cause for it. And if that's true, you can't have some kind of conundrum with us having a particular mutation in the same loci that a lower primate has, because they're probabilistic. That's what's supposed to happen there.
What? Okay, I'm not sure if you know what that word means. If you're like, just based on what you're saying right now, it's not making a lot of sense, T-Rock.
What does probabilistic mean to you?
It means that there's a probability of a mutation happening at each individual spot. Like it's happening probabilistically.
I don't think I would take it quite that way, because that's too generic.
That's literally what it means.
What I would say is that, for example, if you get exposed to asbestos, if 10 different people get exposed to asbestos, the mutations that you suffer are going to be very similar in all 10 individuals.
No,
because
that's not how that's not at all what he's meant by probabilistic.
And you're saying what he meant by probabilistic is that any given loci has a probability of having a mutation?
Yes.
That's completely meaningless, though. That's the same as a random.
Yes, that's how he was using it.
No, he specifically said they're probabilistic, not random.
Yeah, because if you're saying that they're random, that means that each individual loci has the same or roughly the same well, it would be the same probability of mutation. But if you're saying it's probabilistic, then one loci can have a higher probability of mutating than another loci. So you have hot and cold spots. So it's not exactly random, it's probabilistic.
So I think my point still stands, though, because whenever you get subjected to environmental influences of any kind, whether it's chemistry or stress or whatever the case may be, to me, it's largely just simple chemistry. But if you're subjected to the same chemistry, for example, if you're drinking from the same pond water that the animals are, and it's got lead in it, then to me that strongly signals that you all get roughly similar mutations from
That's just not true, though. That's in reality, that is not the case.
Except it is the case, because that's why they tell you don't smoke during pregnancy. That's why they tell you don't take certain medications, because it is known
Yeah,
to
but I don't
cause
think that
this
you're actually
particular effect.
understanding. I don't think you're actually understanding what's going on with those warnings. So, like, the reason why is like every single time you smoke, the molecules of the smoke, once they're metabolized, become sticky to DNA, and so they stick onto your DNA in a probabilistic location, right? You're just rolling a dice every time you smoke. They're going to stick to your DNA at some location, and you just hope that that location is not important. So there's a probabilistic chance every time you smoke that some of those particles are going to stick onto important parts of your DNA that you need, and it will become cancerous mutation. So
So,
that's what is meant by probabilistic. It's
okay.
not like it's the same place every time you smoke. It's it's probabilistic. It's like basically random at where it sticks onto your DNA.
I think I was clear. I'm not claiming it's the exact same place, but I'm not imagining it either. That you see it all the time. You see these commercials pop up that say, if you've taken such and such medication and you've experienced these specific symptoms, then you have the basis for a legal lawsuit. But the point is that they're identified by the specific effect that some medication has on everybody. Like everybody has almost the same reaction to it.
Yeah, now you're just confusing side effects with mutations.
No.
Yes, that's exactly what you're doing.
No. Here's the thing about the whole discussion about DNA, though. It's not simply for the sake of ancestry. It also has to function in your body to play a role as individual cells that each have their own functions.
What? What's your point that you're trying to make here?
My point is that I've already said it. If you're exposed to the same environmental chemistry that an animal is, and for example, you have lead in the water supply, and it affects your liver functionality, for example, it's going to affect them the same way it does you.
I want to add this point. There's several papers, and there's several really detailed papers where they go through the phylogenetics of the issue and all the different species in the biological world that have lost their ability to synthesize vitamin C. And a lot of it has to do with common environmental stressors and triggers and reasons, basically. This one is just a simple breakdown in the abstract, but they point out here we review the genetic bases behind the repeated losses in the ability to synthesize vitamin C and their implication. They also point out that this ability has been lost. That specific gene has been broken in a number of bird species, bat species, guinea pigs, primates. Obviously, the argument is us, fish, so on and so forth. So my point is there seems to be a lack of purifying selection on this gene, probably because we all live in a world where we can get a lot of vitamin C from our diet. And so over time, that gene is not conserved. It's not subject to purifying selection because, okay, it gets hit with the deleterious mutations. Well, we can still survive. We can still have progeny, and we don't necessarily need that. It just means we're going to have to take more vitamin C from the environment. But the reason why our GULO pseudogene would be broken in a similar way to the chimpanzee GULO pseudogene than, say, a bat or a fish or a guinea pig, I think is pretty obvious, which is because humans and chimpanzees share more in terms of morphology, anatomy, physiology, and genetics anyways. And so our GULO pseudogene probably looked very similar at creation or at the flood. It looked more similar than if we were to bring in, a bird GULO gene. Okay, so yeah, it's going to look more similar. And so it's going to break in a more similar way than a bird GULO gene broke. Does that make sense?
No.
Well,
Why
why?
do you think that the pattern of the breaking is going to depend on what? The diet, the function? What are you saying? Why would the pattern of breaking depend on any of that? Like the actual location in the gene where the mutation that breaks it occurs. Why would that be shared
Because
across different created kinds?
that's a fair question. Okay, let's say you have at creation, but I think we should start with the flood. If you have humans and then whatever the chimpanzee bonobo archetype was, okay, if their gene was still intact, if it was still producing vitamin C, as a matter of fact, it may not have by that point. But here's a working hypothesis, okay? Their gene for producing vitamin C would be more similar in genetic content and sequence than the chimpanzee, human, and let's say a pig or a fish. Okay. And so the reason why these genes are subject to breaking down and they're not subject to purifying selection is because of the environment. Because we as humans, we intake a lot of vitamin C in our diet. Chimpanzees do as well. Maybe we intake vitamin C in different ways,
I
but
don't
that
like
doesn't
fruit, so
but that doesn't stop the fact that they're breaking down for the same reason, which is because we're getting vitamin C externally. So
But
that
okay, but you
gene
agree.
doesn't have to be preserved by selection. And so when it breaks in a similar fashion with humans and chimps. But because the gene was already very similar, it also broke in a similar way.
But wait.
I mean, this is the very basis for practicing medicine on mice before you stick it in people. It's because they
No,
do.
it's not. Okay, wait. You agree, Donny, that there are multiple locations in that gene where if a mutation happened in that location, that gene would be broken. There are multiple locations where that is the case. So why is it that between different created kinds, that the location where the mutation occurs is the same?
Because of mutational hotspots and also areas in the genome that are not subject to purifying selection, such as the GULO pseudogene. We're calling it pseudogene now because it's probably broken, right? Before it was just a gene, a working gene. And so it's breaking in a very similar way. But I've got a whole slideshow here and I'm trying to find it. Feel free to speak as I find it, where your favorite geneticist, Dr. Jeffrey Tompkins, he looked at primate GULO pseudogenes, and they're not identical in sequence. There are some differences there. You know, they're not just 100% similar. And so they could have broken in slightly different ways, but still in more of a similar way than, let's say, a pig or a fish gene for synthesizing vitamin C. Go ahead, speak to that a little bit while I find this.
Well, yeah, I think you're making my point is that, like, when you do a comparison across multiple species, the similarities shake out in a nested hierarchy predicted by evolution, even for like broken pseudogenes or ERVs or like lots of these more unconstrained regions of the genome. That's the whole point that we've been
I
making this
doubt
whole
it.
time.
With vitamin C, I doubt it. You might have a hierarchy within, you know, let's say the great apes. If you want to include humans, you would say humans are a human great ape. So you might have the vitamin C pseudogene is more similar between humans and the great apes than between any other creature. But once you get into like fish and bats and guinea pigs and pigs, we don't know if there's a perfect hierarchy there. It could be all over the place. What do you think? There might be a hierarchy within primates, but we don't know that that extends into the entire biological world.
Yeah, I find it pretty unconvincing that like the GULO pseudogenes would be broken in the same spot in every species.
They're not broken in the same spot in every species.
Then if they're broken in different spots for the primates like us versus the other species, then that would be a nested hierarchy pattern. Because
But again,
we would be more similar. Our GULO genes would be more similar to the other primates in the breakage and less similar to more distantly related species in the actual breakage, not the actual like sequence of the GULO genes. We're just talking about the mutations that have occurred in that gene.
I just feel this argument is, it's kind of a desperate argument, because we understand, I mean, right here on the screen, degradation of the GULO gene in a wide array of animals or mammals and birds. It's a common occurrence. So who cares if our GULO gene is broken in the same way as a chimpanzee GULO pseudogene? It what does that prove? It proves that both genes broke, and we both live in the same world, and we both intake vitamin C externally from the environment. And so if other creatures in the biological world experience their own independent breakages, then why couldn't humans and chimpanzees? It seems agnostic to in the wintertime.
No, okay, look, it's an exact example of a nested hierarchical pattern in a non-constrained gene region.
But again, the human and the chimpanzee share more already in anatomy, morphology, physiology, and genetics than a human, a chimpanzee, and a fish. And so of course, that pseudogene is going to look more similar between a human and a chimp than if you bring in a pig or if you bring in a fish. I don't see why this is a problem. Before you speak, T-Rock, and then you speak as well, Grayson, this is Dr. Tompkins' analysis. I don't know how accurate it is, but he points out that when he compared the chimpanzee and gorilla, you know, the certain the 28,000 and so on bases of GULO region is only 84% and 87% identical to chimpanzee and gorilla. So he's pointing out at least that it's not like we have this identical GULO pseudogene between humans, chimpanzees, bonobos, gorillas, orangutans. It's not like it's exactly the same. There's some differences there. And maybe that's because of separate ancestry. Go ahead, speak to that a bit.
So if we stop talking mutations for a minute and talk instead about dietary common sense, you can kind of analyze this from the opposite end of the spectrum. Instead of talking about mutations and genes that are broken, you can instead say, okay, if I can eat vitamin C and get a benefit from it, and a chimp can eat vitamin C and get a benefit from it, and a rabbit can eat vitamin C and get a benefit from it, it's obviously that the chemistry of vitamin C is doing something very similar in all three organisms, despite them being vastly different to each other. It can't be fundamentally different than how deleterious mutations work. It's all chemistry. And so to me, it's the fact that you've got some sort of perceived hierarchy in broken genes, it's got to follow suit that our food supply does the same thing for us that it does for the other animals.
Yeah,
in a beneficial way.
How is that relevant at all to what's being discussed?
Because instead of talking about fundamentally bad mutations, what we're saying is, in order to build a healthy cell has its DNA arranged properly. In order to build a healthy cell, you eat healthy food with healthy chemistry, and it does the same thing in that direction. And why wouldn't it do the same thing in the opposite direction? If you substitute a healthy diet for an unhealthy diet and break something, it should fundamentally follow the same pattern.
No, that does not work, man. That's not how any of this works in real life.
Yes, that's exactly.
But Grayson, notice this. It seems that this GULO gene degradation, the one found in humans and chimpanzees, it occurs, we already know this, we agreed, frequently in mammals and birds. But looking at the genotype, looking at on a genetic level what's going on here, we get it. They're not producing vitamin C. Okay. What's going on with the details of the GULO gene itself? And the events typically are associated with complete or partial loss of exons, these protein coding regions here. So this indicates that the gene may be predisposed to the effects of genetic degeneration. And so if it's the same thing genotypically that's occurring widespread in the animal kingdom. And within all those creatures,
I want to address this.
Yeah, go ahead.
Okay, so again, this is obviously a biased source. We know it's from answers in Genesis, but moving beyond that to what it actually says and what it leaves out, which is very, very important that it leaves out here, is the pattern of breakage. How is this breakage actually occurring? Where are the mutations that are causing this breakage in the GULO gene? They completely ignore that.
No, he goes into it in detail over here. I showed you the 13,000 bases upstream of the human GULO region corresponding. He goes in, it's only 68%, 73% identical to chimpanzee and gorilla. He goes into the 6 exon regions. Have you read this specific paper at all?
How is that addressing it, though?
Because he's showing that all the GULO gene, also the bases upstream, the bases, around it in general, they're not like they're identical. It's because the way you evolutionists frame this argument, it's like, okay, we've got homology. You got this pseudogene in humans, pseudogene in chimpanzees, and it's broken in the exact same way. It's literally identical. It's almost like if we had 2 essays and there was an identical mistake, on page 3 in one and page 3 in the other. That's how you're framing it. But when you actually look at it and all the details on on the genotype, then there's also differences. It's not like it's broken in exactly the same way. It doesn't
none
look
of this
identical.
is saying that, though. I feel like you're equivocating what's on this screen right now to saying that they're broken in different ways, which it absolutely doesn't say anything of the sort.
Are you saying
they're not talking about the mutations that broke the gene in this analysis?
Right, he's talking about the bases upstream, the surrounding region,
yeah,
all the details of what's going on in.
So they're not talking about what where the location of the actual mutations that cause the breakage are. They're just obfuscating it by talking about other metrics of just looking at the total similarities. Nobody's saying that these are identically similar in the whole gene sequence.
Right.
What's similar is the location of the mutation that caused the break.
I just find it funny that now, after we've discussed the unconstrained regions and the hierarchies that supposedly shouldn't be there if young Earth creation is true, now we're just focusing in on just one pseudogene that may or may not be representing a broken gene. It probably is representing a broken gene, or maybe humans never actually had a gene to produce their own vitamin C, because again, from the young Earth creation starting point, Adam and Eve are thrown into a garden with plenty of fruits, plenty of vegetables. You got the seeds as well. And so why do they need a gene to produce vitamin C? Maybe that gene is there for another function, and maybe whatever that function was, maybe that's been degenerated over time. I don't know. That's a prediction. Maybe it needs to be tested. But worst case scenario, even if it is a broken vitamin C synthesizing gene, who cares? All kinds of creatures in the biological world experience mutations and degradation of that same gene independently, not even related to ancestry. But the reason why the chimp and human is more similar in its breakage is because they're more similar anyways. It's not a surprise to me, but anyways, go ahead.
Yeah, I just don't think that logically follows. Just because they're similar, if you actually look at the probabilities of where such a mutation that could cause a breakage could occur, you can still calculate the probabilities. It's not going to be 100% that you're going to get the same in each species. And so for each species, you'd have to calculate the probability of that mutation occurring in that location, and then multiply it by all the other species that you have the same breakage location in. And that lowers the probability each and every time to the point where I don't think it's statistically going to be very high that we get the outcome that we observe today.
It's not. It's one gene. I don't know why this is coming down to. It's one gene that produces vitamin C, and it broke in humans and chimpanzees because we're getting enough from the environment. So it broke in the same way. I don't know why this is statistically implausible.
Let me ask a question real quick. Go ahead, Jackson.
Oh, I just said I don't eat any fruit. I don't get enough vitamin C. I was just making a joke. Go ahead.
See, now, Jackson, you would benefit from that GULO gene being turned back on and working for you.
So let me ask a question, because I don't really know the answer, but it's my understanding that like dogs do synthesize their own vitamin C, right? Is that correct?
Not sure.
You're not sure. So I guess my point here, though, I'm going to assume for the sake of argument that it is true, because I think that is the case, that dogs synthesize their own vitamin C, but people don't. So I guess what I'm saying is I would look
Yes,
for
they do. I just looked it up.
Okay, I would look for a trend among omnivores or carnivores to see if there is a trend specific to certain dietary.
If you want,
leanings.
I can show you a paper where it shows that there's no trend among like the diets of whether or not the vitamin C gene is going to be broken. I know of the paper that shows that.
Yeah, if you want to send that to me, that'd be great. What I often find is that the detail isn't quite specific enough to really put it together. Maybe it is. I've never seen this paper. I'm not trying to make a claim about that paper one way or the other. Obviously, I haven't seen it, but my point is that in humans, we've always had a heavy diet base. Our diet's been heavily based on agriculture, which means we eat, wheat and corn and tomatoes and all that kind of stuff. Whereas animals in the wild tend to go different directions. So you can have animals if they maintain a heavy plant-centered diet. I would suspect that they don't have a need to produce their own vitamin or synthesize their own vitamin C the way dogs do, which is exactly what I would suspect with primates. I would like chimps and gorillas. I suspect the vast majority of their diet is plant-based.
Yeah, that's exactly why we can tolerate a loss in our vitamin C synthesis, obviously, because it's being made up for in our diets. So that's why that it's less constrained. That's why the mutation could happen without a huge impact on our fitness. That's why the mutation could reach fixation without having catastrophic effects, because we can substitute vitamin C in our diet. That's why this sequence is not constrained.
According to your model, when did this genetic mistake become fixed?
I don't know.
Because for us, we have, after the flood, 8 people, right? And so let's say the gene broke pre-flood or even post-flood during that first 500 years after the flood, it could easily become fixed where every single human being has it. And now people spread out to all parts of the globe, taking that genetic mistake with them, where today every single person has it. But according to the evolutionary model, that person who would have got that genetic mistake, basically that mutation, where they can no longer synthesize vitamin C, they'd be the ancestor of all people today. For all people today to have the vitamin C pseudogene, basically, for it to be broken, that'd be a near extinction event. That'd be catastrophic.
I would hypothesize about it similar to what you're saying, Donny, but I would hypothesize about the whole brokenness of the gene. Very different. I would say that it's probably in its natural state, because we are intended to have a plant-based diet, and it's probably lost its ability to adjust for a heavy meat-based diet. In other words, it is the way it's supposed to be, but it's lost its ability to compensate for you if you happen to develop a population that's heavily dependent on meat and with a very low dependency on plants.
Well, I know Chris Roop and Dr. Sanford in Grayson's favorite book, Contested Bones, they predict that it was never there for vitamin C synthesis, that it has an entirely different function that's specific to humans. There's been one other creationist. He put out a technical article where he actually suggested what that function might be. I don't have the technical article on me currently, so I can't remember what that function is. But that is a possibility. And from the separate ancestry starting point, if Grayson were to steel man the model, then it does kind of make sense that for humans, at least, maybe we never had a gene to synthesize vitamin C if we never really needed to synthesize vitamin C because we're just getting a ton externally from the environment.
Okay. It's after 1 a.m. my time, so I'm gonna head out. But I will just, for my closing remarks,
Yeah, do your closing.
I will say that if we never had the ability to synthesize vitamin C, then I would need a sufficient reason to explain why humans and the other great apes have the breakage in similar locations for this gene. I would need a sufficient reason. So if the speculation is that this gene was never intended to synthesize vitamin C, I hear what you're saying, that there's predictions that we could find some other function for it. Maybe even some people have predicted what a possible function for it could be. I don't know. I haven't seen that, but I would need to see the evidence. I would need to see, put up or shut up, basically. I would want to see the evidence that this what looks exactly like a vitamin C synthesizer that's broken. I would need to see what the evidence is that it does actually have a function, and I would need to know what that function is. So if there's evidence for
So
that,
before you
I'm
leave,
open to changing my mind.
Before you leave, I want to say one quick thing to wrap up the main topic of tonight. I think I'm going to retitle the show because I think we had a good discussion on unconstrained sequences and the hierarchies they followed into. I want to give you the last word. So I'm going to say something, T-Rock, you say something. No, wait. I'll say something, then Jackson, you say something, then T-Rock, then Grayson. Let's all
Donny,
make it
I'm
quick.
tired, man.
Okay, fine. Before you leave, I'll say one thing and then you can have a last word if you want. But I just want to say, I think whether it does have an unknown function, it was designed for something different, or it's the result of a mutational hotspot, a gene that's prone to breaking down. I think it's agnostic. You disagree, that's fine. When it comes to the unconstrained sequences and the hierarchies, I don't believe it's a challenge for the creationist position. As a matter of fact, if it is a challenge at all, it's more of a challenge for the evolutionary model because you have those long time scales, okay? For the creationist model, we have 4,500 years for those hierarchies to alter in light of mutations in unconstrained sequences. For much of the same reasons that you look to and Dr. Dan look to, it's not going to alter the hierarchies in 4,500 years. Like, let's get real. Mutations will accumulate, but they're not going to accumulate that many in sequences of the genome that comprise billions of a billion plus DNA letters. Anyways, so I don't think it's a challenge. I think it's been answered. That's my last word. Grayson, go ahead. You have your last word. If you wanted to add anything, then you can leave. Then we'll let T-Rock say something, and then Jackson Rowe gets the final last word. Go ahead, Grayson.
Yeah, okay. So I see no reason to assume why we would start with a pattern of nested hierarchies in non-constrained regions. I mean, that doesn't make sense to me. I mean, there's no common design principles really that explain that to me. I mean, maybe in a future discussion, you can try to explain that one to me. I don't get it. But again, as we saw with the GULO gene example that we went through, it's not just the nested hierarchical patterns in these regions. It's the nested hierarchical patterns of mutations in these regions as well. So that was the point of us going off on that side tangent about the GULO genes. Again, I don't think that your little rhetorical trick of trying to switch it around and trying to make it a problem for evolution and not creation, I really don't think that there's much substance there, but beyond the rhetoric, I do think that it's a significant problem for the creation model, and I think that it's in line with exactly what we would expect for the evolution model. So I'm just going to leave it at that. Good night, gentlemen.
Okay, Grayson,
Nice.
thanks for joining. Always appreciate it. You have a good night. Go get some sleep. You got a busy day tomorrow. T-Rock, final words, final thoughts, my brother?
Yeah, I don't think what he's describing is a challenge for the creation model at all. And so we can look to other examples like why do polar bears and Arctic foxes and Arctic rabbits and so on, why do they all have white fur? It's because that's what their environment does to mammalian fur. That's what it does. It's an environmental trigger that causes a specific reaction without regard to their relatedness to each other. There's tons and tons of that sort of thing. That's why, real colorful plumage exists in tropical regions. It's why venom occurs frequently in very hot regions. That's just natural environmental chemistry. So if indeed we're talking about a truly broken gene as opposed to one that was designed that way and we just don't understand what the function is, we do have a very similar environmental cue to the other great apes. That is, that we do have a heavy plant-based diet, and we've never gone dependent on meat as a population whole. So to me, it's just plain common sense. The environment does repetitive things across the animal kingdom.
Rock, appreciate it, my brother. Looking forward to your open mic coming up on ordering in the fossil record. That is going to be epic. I'm thrilled to host an open mic on that. And I want creationists and evolutionists to join. If you're a creationist that holds to a particular flood model and therefore a particular model in terms of explaining the ordering of the fossils in the rock record,
yeah, if
Jackson's, we want to have a good discussion.
If Jackson's there, we know we'll get to talk about shark teeth, right?
We know Jackson's going to be joining. He's going to be camping out until that open mic. Jackson, over to you. Thanks for joining. Final words, final thoughts.
All right. Well, I looked up the vitamin C thing. It turns out prosimians can't produce or can produce vitamin C, where primates can't. I mean, they're prosimians. Lemurs, for example. There are like 100 species of lemurs. Like half of them, at least, probably more, are like solely herbivorous. So I don't see a correlation between the gene being broken and the diet necessarily. If that's what was being argued, maybe I missed something. But anyway, I'm excited for tomorrow, the genetic entropy thing. So I'll be there for that.
Well, technically tomorrow, the 7th, though. So not, since it's 2 in the morning for me, not technically tomorrow, but
you know what I mean? Yeah.
Tomorrow. Yeah.
Right.
The 7th. So I'm looking forward to that as well. Paul Price, give that one last shout out. Paul Price, genetic entropy and the mystery of the genome. So all you critics, skeptics that have been battling us creationists over the last 4 years, I've been battling the critics on genetic entropy for about 4 years straight. Jackson knows. Jackson for about 2 years. Yeah. So now's your opportunity. Paul Price is, I would argue, an authority on this topic. He's written several articles on it that are published at creation.com, and he's even co-authored with Dr. Rob Carter, one of our favorite young earth creation scientists. So, okay, we'll see you all then. I'm pretty tired, so I'm going to go relax. Jackson, T-Rock, thanks for joining.
I guess.
This turned into a fun 2.5 hour open mic. So with that, Standing for Truth is out. God bless all.
Good night.